So welcome everyone to our webcast, identification of candidate functional polymorphisms using trio family whole exome DNA data, presented today by Dr. Kenneth Kaufman from the Cincinnati Children's Hospital Medical Center. Ken has over 20 years of experience studying genetics, and he has been a Golden Helix SNP and variation suite user for the last couple of years. We have over 750 people registered for this exciting event. I'll tell you why I'm excited. Ken is going to show us an advanced analysis approach that utilizes not only the variant calls from the sequence data, but also the quality information to aid in the identification of novel and rare polymorphisms. And as a special treat, Ken will confirm these results using BAM files in Golden Helix's latest product, GenomeBrowse. On your GoToMeeting pane, there is a section to type in questions. Type them in as we go, and at the end, we'll have a Q&A session to address these. Ken, I know you have a lot to go through today, so I think I'm just going to turn it over to you from here.
Okay, thanks, Dan. I appreciate the opportunity to give the talk today. Let me start just by giving you a little bit of background information. For about the last 7 or 8 years, I've been involved in a number of genome-wide association studies, mostly involving lupus. These studies have been very successful for us. We've identified about 50 genes that are associated with lupus. But even when you look at these genes, it only explains about 30% of the heritability. And part of the reason for this is because in GWAS, in most cases now, you're using common variations, and you're going to be finding the common associations that's very much population-based. It's thought that a lot of this missing heritability is caused by rare or novel polymorphisms that you're just not going to be able to detect with the GWAS type approach. And so one of the things that we're doing to try to find some of these missing genes is taking kind of a personalized genomics approach where we can look for rare variations or novel variations in a very individually focused manner. And the way we're doing that is using trios where we're looking at a mother, father, and a child that is affected with the phenotype that we're interested in. And so just briefly, I wanted to kind of go over what we're going to talk about today, some of the points that I think is important. I think it's good to have an understanding of next generation DNA sequence, what the data is like, what the benefits are, what the pitfalls are, so I'll speak a little bit about that. As I said, we're going to be using a family-based study design, so I'll talk a little bit about that and kind of the strengths of that type of an approach. I think it's very important that before you start generating the data, you really define the experiment, and as well as define the type of polymorphisms that you're hoping to find. And then, obviously, hopefully we'll get to the point where we have some candidate functional polymorphisms that actually cause the phenotype that's being studied. So our goal today, hopefully what I'll be able to show you, is that we can reduce the number of variations found in the exome to a very manageable number. Normally, when you do the whole exome sequencing, you'll find about 50,000 variations in each individual. This corresponds to about 100,000 variations within a trio. And so what we want to do is get down to a very small number of candidates that are likely causing that phenotype. And this just shows you kind of a graphic of a trio. It's a variant map that shows about 115,000 variations found in a trio. This is what you're starting with, and hopefully what we'll get down to is something that looks like this, where you have just a few variants that are most likely to be your phenotype-causing variants. And so the way we're going to try to reduce these number of variants is using a filter-based approach. We're going to filter the variants based on the family structure. We're going to filter the SNPs based on the functional potential of those SNPs, in that they have the ability to somehow alter the protein sequence. And then, as Dan mentioned, we're also going to use the data quality to filter out the results so that we can get rid of sequencing artifacts and bad reads and are left with what's the highest quality data and is most likely real results. Now, with next generation sequencing, there's some things that need to be taken into consideration. There's multiple platforms that can be used to generate this data. Most of these employ short reads of about 100 base pairs. They generate millions and millions of reads, and then those reads are aligned to the reference genome. And then any place where it's different, once you've aligned it to the reference genome, that's considered a variation, and that's what we're looking for. Now, there's a number of issues with next generation sequencing. These first four here, these are going to be more the responsibility of whoever's generating the data for you. But one of the things I would like to point out is that there is a very large bioinformatic burden in these type of experiments. And when you're actually analyzing the data, this can become an issue also. The thing that we're going to be interested in are sequencing artifacts. These are places where variation is called when it's not really true. And these can come about because of just bad sequence data or poor alignment, areas that got aligned to the genome wrong, and that caused a variant to be called that's really not there. There's other issues, but these are some of the things that you need to take into account. One of the things that you'll hear a lot of times with next generation sequencing is coverage. And coverage refers to the number of reads at any one particular base. And so usually you'll hear something like 30x coverage, 50x coverage, 100x coverage. The higher your coverage, the better your data is going to be. But the thing that you have to realize is that this is an average coverage, and that you're going to have areas where you have extremely good coverage, but you're also going to have areas that have very low coverage. The other thing to keep in mind is that depending on which platform you use, you'll see differences in the coverage. So when you're trying to do an experiment like this, I think it's important to define the problem. In most cases, it's pretty straightforward. You're going to try to identify functional polymorphisms that lead to a phenotype. Then I think this is a critical step. You need to try to determine the possible genetic mechanisms of disease. What is it that you think is actually causing your phenotype? It could be polymorphisms within the promoter or other regulatory elements. It could be some type of polymorphism that alters the coding sequence of a variant or a splice site. Depending on what model or what you think the mechanism really dictates how you design the experiment. There's a number of options. You can do a whole genome sequence, which you might want to do if you think that it's going to be a promoter or some type of regulatory element. If you think it's in the exome in an exon of a gene, then an exome sequence approach could be appropriate. If you're not sure but you have candidate genes, then you might want to do some type of targeted sequence capture approach where you're capturing both the exons, introns, as well as flanking sequence of specific genes for analysis. I think, it's really important to realize that if you're doing exome sequencing, then you're not going to be able to find polymorphisms in a promoter region or intronic sequences that have the potential to alter the expression level. So it really needs to you need to have a clear idea of what it is that you're looking for. And so, as we've said, we're going to be working with trio studies today. This is a father and a mother with a child that has the affected phenotype. In our case, neither mother or father has the phenotype. The advantages of this is that it's a small sample size. It's an N of 3. The bioinformatic burden is going to be much lower. And you also have some built-in QC. You can look for Mendel errors within the samples. In fact, we even use the Mendel errors as one of our filtering criteria. Another advantage is that you have phase. When you look at the polymorphisms in the child, you can determine if that polymorphism came from the mother or father. And this can be beneficial in trying to understand the phenotype. It is a very personalized approach. Hopefully what you're going to be able to do is identify what's causing the disease in this particular child. And it's ideally suited for whole exome sequencing. Now there's three types of polymorphisms that we're going to talk about today for the analysis: de novo, compound heterozygous, and homozygous recessive. I'm going to start with the de novo polymorphisms. These are polymorphisms that spontaneously occur in the child. Neither parent has this polymorphism. So by definition, this is a Mendel error. These are, like I said, a non-inherited DNA mutation. There's multiple mechanisms that this can occur by. We think that this polymorphism has to occur relatively early in development so that the signal-to-noise ratio is above background. If it's only occurring in a small percentage of the cells, then we're probably not going to be able to detect it with this technology. We think that in most cases, these de novo polymorphisms will be unique to the patient. However, there has been some studies come out recently showing that the same de novo mutation is occurring in multiple patients, which I find extremely interesting. We expect that this polymorphism is going to be heterozygous in the child. The chances of having two de novo polymorphisms at the same site would be extremely rare, so we do expect the child to be heterozygous for the polymorphism. And as I said, we also expect that this polymorphism will have the potential to somehow alter the amino acid sequence of a protein. It could be a non-synonymous polymorphism, introduction of a stop or loss, the gain or loss of a stop codon, change the open reading frame, or alter splicing of a protein. So the filters that we're going to use today is, first of all, that it does alter the amino acid sequence, that it's relatively rare. And in the example today, what I'm going to use is data from the exome sequencing project with the criteria that the minor allele frequency of the SNP has to be less than 1%. This will also include any novel polymorphisms. By definition, a de novo SNP is a Mendel error, so we're going to use that as one of our filters. And as I mentioned, the child has to be heterozygous. And then we're also going to use some quality measurements on the genotypes to remove the sequence that's the least reliable and most likely a sequencing artifact. And these measurements are allele depth. This is the number of reference alleles that were detected for the SNP and the number of alt alleles. The overall read depth, which is just how many reads were over at that particular polymorphic site that were used to make the genotype call. And then what's called the genotype quality score. And this is a score that's given that estimates how good a genotype was made. What we work with is VCF files, and these three different quality measurements are listed in the VCF files, AD, DP, and GQ. Now, if you ever look in one of the VCF files, you'll see that there's actually a lot of information about the quality of the genotypes, the reads, and these can all be used for filtering. What we've found is that these three filters right here are the best indications for filtering out low-quality sequencing data, but you can definitely use these other measurements for filtering if you want to. And so why is this important? This is a histogram of the coverage of a typical whole exome sequencing project. In this particular sample, we got an average of about 50x coverage, which you can see is in this region here. So most of the sequence is going to be extremely high quality. However, when you look at the Mendel errors, you can see that the coverage is skewed. And in this particular case, with the Mendel errors, the average coverage for Mendel errors is about 10%, or about 10x, which is an indication of the low quality of the scores. So when you're looking for de novo mutations, you're basically going to be going through the lowest quality reads and sequence data to try to find the real de novo polymorphisms. Okay, so let me switch over here to Golden Helix. This is SBS software. Now, I've already imported a few of the files into this project just to save time. One of the files is the pedigree information. The VCF file I'm going to be using today has 2 families, and so this just has the family ID, patient ID, father, mother IDs, and gender and the status. And I've also imported the VCF file, and that's done under the Import tab, Import VCF from one or more files. I'm going to show you what this looks like. But when you import it, you get the option to scan the you have to scan the VCF file. And what that does is it shows you all the information that's in the VCF file, and you have the ability to select what you want to import into your SVS project. And so in the case of today, we're going to use the allelic depths, the approximate read depths, and the genotype quality score. But here's all the other quality measurements that you can also import in and use as filters if you want to. One of the things that's also nice about this is you have some control over how you import the data. You can import it into the marker map, into a spreadsheet, or you can do both. I'm going to go ahead and cancel out of this because I've already imported this file. When you import the VCF files, you get a genotype file, spreadsheet, as well as the allele depths and the quality and the scores and the overall depth. And I'll talk about those a little more in general. The other thing that I've already done is did the variant classification on these genotypes to save some time. That's done under Analysis, Variant Classification. And when you do that, it generates a number of spreadsheets. The one that we're going to be interested in is the coding variant classification. And what you see is that when we started off here, we started off with 113,000 variations in these 2 trios. When we look at the coding variation classification, 42,000 of those variations are within a gene. We can further reduce this by activating by category, where we say we're going to look at everything except for the synonymous SNPs, because those won't change the amino acid sequence. And so when we apply that to it, our list of variations goes from 42,000 to 22,000. This is kind of a typical number what we see when we look with trios. So I'm going to go ahead and make a new spreadsheet and rename this so I know what it is. I'm going to go back to my genotypes and just activate the non-synonymous SNPs. We're going to do that by using our subset non-synonymous SNPs here. So here's our 22,000 SNPs that are active. At this point, I'm going to go ahead and add the pedigree information to it. And rename it non-synonymous so I know what it is. So now we have this spreadsheet that has the genotypes as well as the pedigree information. And at this point, I'm going to go ahead and filter out based on the frequency of the SNPs. And as I said earlier, I'm going to use the exome sequencing project. I'm going to use European American minor allele frequency that is less than 1% as my criteria. And so what you can see is that of those 22,000 SNPs that we started with, about 8,000 of them would be considered rare. I'm going to go ahead and create a top-level spreadsheet here. So we've already gone from 113,000 variations down to about 8,000. I'm going to now use a new script that's actually going to subset this spreadsheet into multiple spreadsheets based on a criteria. The criteria I'm going to use is the family ID. This is a new script. It's actually very nice when you have DCF files that have a large number of samples in them. It's called Subset Informative Genotypes by Category. And what it allows you to do is select out whatever it is that you want to subset by, in this case, family ID. And what it'll do is it'll go through and read this spreadsheet and generate two new spreadsheets, one for each family. Or it'll generate a spreadsheet for each family. So again, I'm going to rename this just so I know what it is. So if you remember, what we're looking for are de novo SNPs. And so by definition, a de novo SNP is going to be a Mendel error. And so we can run the Mendel error check on this family. And we find that of these 6,000 SNPs that we're down to, there's 385 Mendel errors. And then this spreadsheet here will actually give us a list of those, of all the SNPs. And if you remember, one of the filters that we're going to filter on is not only that it's a Mendel error, but the child is heterozygous. So we're going to come over here and activate ones, so that this will activate where the child is heterozygous. And then come over here and activate by a threshold where it's also equal to 1. Check this box here, so the filter is only going to apply to the activated samples or SNPs. And generate a new spreadsheet here where these are the Mendel errors where the child is heterozygous. So at this point, we're down to 305 candidates. And so now we can begin to filter based on the quality of the calls that were made for these polymorphisms. And one of the things I like to do is rename these to something that's a little bit shorter and something I can easily recognize. So the first one is the number of reference alleles, then the number of alt alleles or alternate alleles. We have the genotype quality score, and then the depth. So let me go back here. So what I'm going to do now is activate just the SNPs that are Mendel errors and that are heterozygous. I'm going to reactivate the family structure. Go ahead and create a top-level spreadsheet just so I can keep track of it. So this is family 1, and this is going to be for de novo candidates. Okay, I'm going to go ahead and use that script again that will subset this for me based on the family ID. Rename this to family 1. And so what I'm going to do now is also use a new script that's going to generate a new spreadsheet that has all the information from the reference alleles, alt alleles, quality, and depth. For my own personal preference is to go ahead and recode these genotypes so that it indicates if it has the alternate or the reference allele. This will make it a little bit easier when we're filtering later on, because all of the genotypes are in a similar format as opposed to having the base genotypes. And so now I'm going to generate a spreadsheet that has all the quality information with those genotypes. And so I'm going to use this script called build sample collated spreadsheet. One of the things that's nice about this script is the order that you click on your spreadsheets will be the order it appears in your output. I'm going to give it a name so I know what it is. You have the option of saying what the headers are going to be. And so this script will actually take you through about 30 steps that you'd have to do manually otherwise. Now, if you look at this data, the spreadsheet, the way it's set up is you have the genotype, the number of reference alleles, alt alleles, the overall depth, and the quality score. This first sample happens to be the patient. I like to activate it just so I can keep track of it. And so now we can begin to filter based on these quality measurements. In our experience, what we've seen is that the alt alleles and the reference alleles is a good indication of the quality. What we use as a cutoff for the parents for the alt allele is that there can only be one or no calls for the alt allele. You see that takes us from 305 SNPs down to 96 SNPs. And come back and apply the same filter to the other parent. It's going to take us down to 70 candidates. We can also filter on the depth. The setting that we're using right now is that the depth has to be equal to or greater than 15. Takes us down to 48 candidates. Apply the same filter to the other parent. Down to 37 candidates. And then we can also use the genotype quality score. And what we're using is 20. Now these filters that I'm applying are not really that stringent, but they do a good job of removing the low quality sequences. And we're going to apply that filter to the last parent also. Okay. So that's taken us down to 32 candidates. And if you'll notice, I've not done any filtering based on the patient at all as far as the quality of their reads. One of the things that we've found in the de novo SNPs that we've validated is that we have not found any real de novo SNPs that had an alternative reference allele less than 10. So we're going to go ahead and remove all of the polymorphisms that have an alt allele less than 10. And that gives us a total of 7 candidates. Now when you look at the list of candidates, you'll notice that we have a number of these candidates that are all coming from chromosome 2 in the same region. This is usually a sign of some type of artifact, usually caused by an insertion deletion. As Dan mentioned in the introduction, they're coming out with this new tool called GenomeBrowse, and we can use that tool to actually visualize these candidates to further filter out what looks like it might be a sequencing artifact or to prioritize our candidates. Now I've already put these SNPs in the format that's needed for this tool. And let me show you how this works. So what I've done in GenomeBrowse is I've loaded the BAM files. BAM files are, have all the information as far as all of those millions of reads that were generated and where they line up in the next genome. And with this tool, you can zoom in and out on the alignments to look at issues like the coverage, which is indicated by this histogram, as well as the alignments you can see here. And in this case, I've got everything sorted by the strand also. So I can tell how many of these sequences correspond to the top strand and how many of these correspond to the bottom strand. And when I look at this particular SNP, this candidate, I see that we have very good coverage. Also, we see that we have representation of the polymorphism on both strands. If you look at where these sequences begin and end, you can see that that varies. That's also another good sign that this is good sequencing data. This top sequence here happens to be the child. The two bottom alignments here are the two parents. You can see we don't see really any indication of this particular polymorphism in the parents. So this would be considered a very good candidate. Let me show you another one of our candidates. If you remember on chromosome 2, I said we had a number of polymorphisms that were showing up as candidates. And when you zoom out on this area, you can see that in the patient, there's actually a number of polymorphisms showing up in this area. We're not seeing those in the parents, but we are seeing something in this parent here. And what it turns out is that this parent actually has an insertion deletion at this particular site. And insertions and deletions cause a lot of problems with the alignments and a lot of artifacts, and that polymorphisms will be called that are just due to a misalignment. And so actually, it turns out that because of this insertion deletion, the child actually has the insertion deletion, but because the sequence quality was either a little bit lower, the coverage wasn't higher, it made a number of false calls for polymorphisms. And so whenever I look through these alignments and I see an insertion or a deletion in one of the parents, that's usually a very good sign that what you're actually looking at is a sequencing artifact. Okay, and let me just go through one more just to show you what GenomeBrowse can do for you. So this is another candidate on chromosome 10. Again, this is where the de novo candidate SNP is. For this particular de novo polymorphism, you can look at the ratio of the alleles. You see it's not very high. That's, again, another sign that it's probably not a real polymorphism. You can see there's two heterozygous calls nearby where the ratio is much better. We're not really seeing anything in the parents, but if you do notice, there is another insertion deletion right by this particular region. And so, again, this is probably going to be a sequencing artifact that's caused by the presence of this insertion deletion. And so one of the things I'd like to really stress is that what we're doing is identifying candidates. We think it's very, very important that these have to be validated. And the approach that we're using to do this is by using PCR to amplify the region that contains the candidate in the child as well as the parents, and then using Sanger sequencing to validate it by another method. And if you remember that first candidate I showed you that looked very good in GenomeBrowse, and the data, everything looked good, when we came back and sequenced that particular SNP, you can see that the child here is definitely heterozygous at this position, and neither parents have the alternate allele, that they're homozygous. So this is a real true de novo SNP that we found. It is within a gene. It does change the amino acid sequence. Okay, and it turns out that when we validated those other candidates, the other insertions and deletions that we found in the parents were actually also in the child, and they were causing those to be not real. So now the next type of polymorphism I want to talk about is compound heterozygous polymorphisms. These are going to be polymorphisms where the father is heterozygous at one position The mother's going to be heterozygous at a different position. Both of these polymorphisms are going to be in the same gene. The child inherits both polymorphisms, and the net result is that both copies of the gene can be potentially affected. So the filters I'm going to use for this type of analysis is that the parents have to be heterozygous for at least 2 different polymorphisms in the same gene. The child's heterozygous for at least 2 of the SNPs, 1 from each parent. The polymorphism, again, alters the amino acid sequence. It's rare, less than 1%. And then we're going to also use the same type of quality measurements in that the genotype call depth has to be greater than 15. The genotype quality score is greater than 20. We're also going to apply a requirement that the heterozygous alt allele has to be greater than 10. These are basically the same measurements I used for the de novo SNPs. So let's go through and do that. Okay, so we've already done some of the filtering. We've already identified which SNPs have the potential to change the amino acid sequence and which ones are rare. Go back to this guy here. So in this family, that's 6,000 SNPs. There's a script that we can run that will score the compound heterozygous regions for us. However, what we really want to do is do all of our filtering first so that we have as small a list as candidates as possible. So what I'm going to go ahead and do first is use the script here that's going to collate my quality measurements with my genotype data. It's going to be for compound hets. So this is the same spreadsheet that we used before. So again, the patient is the first sample, and the other set of columns correspond to the parents. So the first filter that we can apply to this is that the child has to be heterozygous. Oh, we already got that in. Oh, just a sec. I think I messed it up. Let's go back here to... Okay, let me regenerate this here. Okay, let's start back here at the beginning here. Okay, so here's our candidate of 6,000 polymorphisms. Spreadsheets are so we want to add our recoded genotypes and then our quality matrices. Okay, here we go. So again, this first set of data corresponds to the patient. We're going to activate by a category. Now this is the reason why I recoded it so that all of the genotypes will be in the same format. It's easier to filter on the heterozygotes. So all of our patients, or all these SNPs are going to be heterozygous in the patient. We're going to go ahead and apply the filter for the alternate allele, saying that it has to be greater than 10. The quality score has to be greater than 20. And that the depth is going to be greater than 15. So you can see we started off with 6,000 polymorphisms. We're down to about 2,700, 2,800. We can apply these filters to the parents. Now we're not really going to look at the alt allele filter for the parents, because we expect for some of these SNPs, one of the parents will be homozygous and the other one's heterozygous. But we can filter on the overall quality of the genotype. As well as the depth. And we can do this for the other parent also. So it takes us down to about 2,500 candidates. Let me just create a top-level spreadsheet here. And so we're going to use this now to activate our genotypes. We need to remember to reactivate the family information. Create a top-level spreadsheet here. And now we're going to use a script that will score our compound heterozygous SNPs for us. So you can see what we found here was 227 potential compound heterozygotes where the father's heterozygous for one polymorphism, the mother's not, or vice versa. If you look at this list, you can see that actually a number of these SNPs or polymorphisms are occurring multiple times. And that's because they're in different transcripts. And so actually the number is actually a little bit lower than this. It's about 180 variations that's listed on here. Let's see what it's doing. If we look at the genes that actually have the compound hets in this particular trio, there's only 42 candidate genes. And again, if you look at these, some of these genes are listed multiple times. And if you remove the duplicates, we're actually only looking at 36 genes. Now at this point, we can use GenomeBrowse again to look at the quality of the sequencing data to try to decrease the number of candidates. So let me show you what some of those look like. So here in GenomeBrowse, I'm going to look at a SNP that's on chromosome 7. You can see that the patient does look like, or the child does look like they are heterozygous here. We're getting reads for the alternate allele on both strands in different positions. Looks really good. This parent here is obviously heterozygous also, and the other parent's homozygous. So this looks good. If we look at the other polymorphism in this gene, we can see it fits the model that we've described and that the child looks like they are heterozygous. We're really not seeing this allele show up on the sequence that's on the bottom strand, which would make me feel more comfortable about it, but also our coverage for the bottom strand sequence is low. This parent is definitely looks heterozygous. This one looks homozygous. And if you remember from the first SNP I showed you, it was flipped around as far as which parent had the polymorphism. So this is actually a very good candidate. It fits the model and it fits the description of what we're looking for. Let me show you another candidate on chromosome 1. So this first SNP, the child was called heterozygous, as was this parent, but the other parent was actually called homozygous G. But if you look at it, there looks like there's a lot of alternate alleles that correspond to the A allele in this particular sample. So this is probably a miscall, in that both parents are actually heterozygous at this position. If we look at the other polymorphism in this gene, you can see that it's actually very similar. The child was called heterozygous, this parent was called heterozygous, this parent was called homozygous, but we are seeing evidence of the alternate allele in this parent. And so this is actually not a very good candidate because both parents appear to be heterozygous for these particular polymorphisms. So you can use GenomeBrowse to go through your candidates fairly quickly and go ahead and further window out your candidates to get a better list of what's going to be a true result or not. Okay, let's go back to this guy here. And so the last type of polymorphism I'm going to talk about is a rare recessive homozygous polymorphism. These are going to be polymorphisms where both the father and the mother are heterozygous for a polymorphism, the same polymorphism. The child inherited both of the alt alleles from both of its parents, and so the net result is that they're homozygous for the alternate allele. Filters that we're going to apply, very similar to what we've been doing. By definition, we're saying the parents are going to be heterozygous for the same polymorphism. The child is going to be homozygous for that alternate allele. The polymorphism has to alter the amino acid sequence. That they're rare. In our case, we're using a minor allele frequency, again, of 1% based on the Exome Sequencing Project database. The genotype has a call depth greater than 15, quality score greater than 20, and that the heterozygous alt allele is greater than 10. So let's get started with that. Okay. Let's see here where I will start. Again, I'm going to use one of the spreadsheets here where we have already defined our non-synonymous SNPs that are very rare. I'm going to go ahead and create a top level just to keep this separate from everything else. We're going to call this homozygous recessive. We go to genotypes. And we're going to go ahead and add this to our quality measurements. Tools. I call it homozygous recessive quality control. Here I'm just changing what's going to be in the header for the genotype. So again, this is the same spreadsheet that we generated before for these other models. By definition, we're saying that the child has to be homozygous for this polymorphism for the alt allele, so we can activate just those SNPs where the child's homozygous for the alternate allele. And you see that'll take us down from 6,000 to 800 polymorphisms. In this case, we could go ahead and filter based on the reference allele. Since they're going to be homozygous for the alternate allele, then we can get rid of anything that has that reference allele in it. And so in this case here, I'm going to use a filter of less than 2 so that only the polymorphisms that have 0 or 1 reference allele will be counted. That takes us down to 412 polymorphisms. And so now we can begin to filter on the quality scores of the patient. And again, these are going to be the same as that we've used already. 20 for the genotype quality score. 15 for the depth. It's going to take us down to just 78 SNPs or polymorphisms. And now we can begin to look at the parents. And so by definition, again, the parents are going to have to be heterozygous for the polymorphisms that we're looking for. So we can come in here, activate by category, say that they have, this parent has to be heterozygous, and that the other parent also has to be heterozygous. And you see, even at this level here, it's taken us down just to 3 candidates to look at. And again, we can use GenomeBrowse to look at these candidates. We've got some of those over here. So in this particular polymorphism, the child is looking like they are homozygous for this T allele. The 2 parents are looking very heterozygous. We see good coverage. We're seeing it on both strands. There's multiple start sites for the alignment. So this would be considered a very good candidate. Let's go ahead and look at one of the other candidates here. Put the coordinates in GenomeBrowse. Okay, so here's the site that said this was the polymorphism. And what you'll notice again is that there's an insertion deletion in this area. And usually whenever we see an insertion or deletion, it's going to be a sequencing artifact, and it's not going to be a real result. Okay, let me jump back to here. So what we've done today is we've looked for de novo polymorphisms in this trio. We were able to filter the results down to 7 candidates. We removed 4 of those with GenomeBrowse, and experimentally, when we came in with another technique, we were able to validate one of those as being a real compound heterozygous, de novo polymorphism. With the compound heterozygous, we identified 185 variants. That corresponded to 34 genes. However, using GenomeBrowse, we could actually go through and remove 26 of those genes to give us a very small list of compound heterozygous polymorphisms. With the rare recessive homozygous, we had 13 candidates, and I didn't show this, but 7 of those candidates were in the HLA region, which is very polymorphic, and I actually could remove 2 of those with GenomeBrowse. And so what did that do? So we started off, if you remember, with about 113,000 variants. After all of our filtering for these 3 different types of polymorphisms and using GenomeBrowse to further look at the quality of the data, visually inspect it, we end up with about 40 variants that looked like they were very good candidates for causing the phenotype. And if you think about the numbers, that results in a 99.96 reduction in the total number of variations. And so you end up with a list of candidates that's very manageable, and then you can start to use other tools, bioinformatic tools, or known features of these genes to further prioritize your results if needed. And I think that's the end of the talk.
Ken, we can't thank you enough for putting together such a captivating talk. I just want to say a couple of things before we jumped into questions here. One is that GenomeBrowse will be publicly available by mid-September, and it will be free to use. For those of you watching who are interested in applying this kind of technology to your research, just let us know. When you exit the webcast after Q&A, there'll be a survey, and you can check the box to be contacted, or just give us a call. We'd be happy to set up a simple discussion with some of our technical staff and talk through your current and upcoming research goals while exploring how Golden Helix and SVS may be able to play a role. You can also go to the website to learn more about SVS and GenomeBrowse and sign up to be notified when GenomeBrowse becomes available. With that said, I'm going to start going through a couple of these questions, Ken, if you've still got it in you.
Sure.
We had a question. How much harder would this problem be if this was whole genome sequence data rather than whole exome sequence?
So I have not personally worked with the whole genome data, but the filters, so I can't say exactly how these filters would apply to the number of candidates that you would get, but I think from our experience, you know, it does give you a list of the results, the variants that are real. Now, you know, when you look at the data that you get back from next generation sequencing, the vast majority of it is going to be high quality and is going to be accurate. The places where you're going to have errors in that data, those are going to show up with variants. So by default, you're kind of only looking at a very small subset of the data that has the highest potential to have sequencing artifacts in it. And so if you're looking at whole genome, your overall numbers are going to probably increase. You know, I think if that's the case, then you would probably either have to you know, increase some of these filters, for instance, depth or the genotype quality scores, to try to winnow out some of the bad calls. The approach we've taken is actually not to be that stringent, or at least we feel that it's not that stringent with the filters that we're using, because we don't want to miss a potential real result just because the coverage in the area was bad or something like that. But we also don't want to have a lot of false positives. And for us, this gives us a pretty comfortable window. Right now, with like the de novo polymorphisms, about 40% of our candidates are validating as being real. As we continue to do this and generate more data, we're constantly fine-tuning these filters as well as some of those other quality control measurements. I would think with the whole genome, you would probably have to use some other filters to try to get your numbers down, though.
Excellent, excellent. And I should mention, we're at the top of the hour, and I just thought I'd let people know who may have things on their calendar right after this talk. We're going to keep going to get through these questions because there's a lot of actually real interesting ones. So hang on the line if you're interested. If not, don't feel bad about leaving early. Can we have another question? What reference genome did you align your data to? And have you ever encountered a situation in which the reference genome is incorrect?
So in this particular data set today, I kind of cheated. This is actually a little bit older data set. It was originally built based on build 36, and that's what the BAM files that I was using. But shortly after we got this data set, we started using build 37. And so we recalled this data, but for the talk I did not actually have the build 37 BAM files, and so that's why I was kind of using that cheat sheet, because those were the candidates had been already transposed back to build 36, so I could use GenomeBrowse to find those sites very quickly. I have not really noticed any differences other than things that you typically see when you go from one build to another build or reference to another. You know, there'll be, when we use build 36 data, for instance, there'll be polymorphisms that were detected, and then when you look in build 37, those regions have been deleted out of the genome. And so does that mean that build 36 was wrong or right? I really can't say. You do see some differences. We've noticed that depending on what reference sequence you use, there can be a 1 base pair difference in the positions of SNPs, and so that can cause problems, mostly though further down when you're coming back trying to validate these and you want to get the genomic sequence. But as far as with the analysis, I really haven't seen any issues.
Excellent. How do you distinguish between de novo mutations and germline mosaicism?
Well, so again, that it kind of we're calling it de novo polymorphisms, and that's basically what I kind of talked about earlier. How what you really need to do is define what is the type of polymorphism that you're looking for, and so the model that we've put up with the definition of it being heterozygous in the child, the parents have to be homozygous. It's a Mendel, all these filters basically designs a specific type of polymorphism. We have not gotten with our validated polymorphisms yet to look to see what the ratios of these are, if it is some type of mosaicism or not. We don't know. It's a possibility. I do feel that if you do have some type of mosaicism where your de novo might only be in, let's say, 10% of the cells that you're looking at, we'll probably filtering out that result. As I mentioned earlier, for the type of polymorphisms we're looking for, I think they really have to evolve very early in development so that the vast majority of the cells are carrying that, because we are using the depth as one of our filters, and if you have a ratio of the alt allele to the reference allele just based on copy number, you're very likely to not be able to detect it.
Okay. Let's see. There's a couple other questions here that I can probably handle. Someone asked if BAM files are loaded into Golden Helix or there is an alignment tool associated with it. So when Ken was working with an SBS where he did the primary data management and filtering, he was working with VCF files. So those have already been aligned and called for variants. The workflow then transitioned to, once he had filtered down to a smaller set of putative variants, he then went to kind of confirm that variation within the BAM file, again, to do a visual assessment to, I guess, see if it's real or not, to see if it actually necessitates further validation. So again, the question was, are BAM files loaded in or VCF files? It's kind of both. VCF files into the SNP and variation suite, and BAM files into GenomeBrowse for that variant confirmation.
Dan, on my screen, what I'm showing here is the GenomeBrowse, and all of this information here is coming from the BAM files with the alignments.
Exactly. Another question, the scripts that Ken used to collate spreadsheets and attach the quality information, we have a question if Ken created these or if Golden Helix did, and yeah, those are scripts that we created. So we wrote the code and shipped the file. Then Ken could access those features just by kind of clicking the button or setting the drop down. So I'm just combing through these questions here. Here's another question. Was GATK local realignment used around indels to reduce false positive SNP calls?
So all of the data that we're working with is coming from actually 3 different sequencing cores, and they actually run the bioinformatics. I'm not exactly sure what the to answer to that is, because I didn't actually generate the data, but they are using, all 3 cores are using GATK and following the best practices that are published by Broad Institute, and then using the, after variants are called, I know they're using the GATK to generate the multiple sample VCF format following best practices.
Excellent. And I actually should mention in regard to the previous question about the scripts, should have mentioned that they're actually available online, and many of them shipped with the software, so you can download and apply the same tools. And I should also mention that Ken influenced the design of those scripts, essentially from us watching him work.
I had a little bit of input.
Could you explain the alt allele greater than 10 filter and what it means, and I guess what it's based on a bit more?
So when we first started doing this, we were actually not filtering on the child at all. And so what we ended up with is, instead of getting our de novo polymorphisms candidate list down to about 5 per trio, we were around 15 to 20. And so initially we were going through and we were validating all of those. And what we found is that if we didn't filter on the child, our success rate in validating de novo SNPs was less than 10%. And so as we started going through a number of these trios and building up a kind of a large database of what was real and what wasn't, we could start looking at some of these quality measurements and say, what can we filter on to try to get rid of these false positives? So one of the things that we quickly noticed is that almost all of the SNPs where the child was called heterozygous, and when we came back and sequenced them and they were actually homozygous, they had a very low alternate allele depth. In fact, of all the de novo SNPs that we've validated by Sanger sequencing, we've never found one that had a alternate allele depth below 18. But so what we could do is, if we said, when we looked at the data, we said, well, if we use 10, we're going to get rid of about 80% of our false positives, and we still would have been able to detect all of the compound, or all of the de novo polymorphisms that we did validate. And so that's where we've kind of set our threshold for that. It's based kind of on experience, and as we continue to do these type of experiments, we keep looking at those measurements to see, well, maybe we could move that up to 15. If we move it up to 15, would we still be able to have detected all the ones that we validated and get rid of some of the false positives or not? And so that's still very much under development. And and like I said, our stringency, I feel, is not that strong right now, but it does get rid of a vast majority of the bad reads. And one of the things that I would point out is that's one of the things that's extremely powerful with this software package, is that it gives you a tool set where you can actually come in and filter it based on the criteria that you want to. And you have the option to filter on different variables at different levels and see what those results are. And so that's, what we're still in the stage of that, of trying to figure out what's the best way to analyze this data. I mean, I would love to get to a point where we get 100% of our candidates are real and we don't miss anything. I don't think we'll ever get that far, but I think we can probably boost it a little bit. But so that's kind of a long answer to your question.
We've got another one about the clinical application of some of this work. How good is the data, or how good is your confidence with some of this data in a clinical scenario?
So, I mean, that's something that we are looking into. All the stuff that, this particular data I talked about today, and most of the data that, actually all the data we've looked at is based from a research orientation of trying to find candidates to try to understand, maybe identify new genes, identify pathways that could be involved. I think for clinical purposes, coverage is going to become a bigger issue. I think the requirement will be that you'll probably have to be at about 100x average coverage for clinical purposes. The problem is with this technology is that you're always going to have this outlier sequence that is problematic. And so if you increase the coverage and you can cut that away, I think you'll be much more confident in the calls. It's really, I think, kind of too early to say in the technology as far as what are the thresholds that we're going to have to put on the data to say, yeah, this is real. There's definitely filters that I could apply to these data sets where I would feel like the calls are 99% accurate. The problem is going to be is that you then have a very high likelihood of missing stuff. So I think those are issues that we're struggling with and other people are struggling with, of how to work with this data. But I think there definitely will be clinical uses for this type of analysis and experiments.
Okay. We've got another question about population filtering within SVS, and I'd be happy to take that one. Early on in one of Ken's workflows, I believe you filtered out common variants based on allele frequencies from the 1,000 genomes population. So there's a couple of ways to filter using larger databases like that. We can use something like the 1,000 Genomes project or the exome sequencing project, 6,500 exomes data, to filter based on variant frequencies. We can also use resources like HapMap or dbSNP. For example, you could use dbSNP to simply make a decision that, hey, if it's in dbSNP, maybe it's too common to even be considered for whatever disease you're researching. So we do actually have those tools built right within SVS. Let's see, let me pick another good one for you, Ken.
Dan, can I have one other comment to that?
Hey, no problem.
Because one of the things that we do is you can also generate your own database and filter based on your own custom database too.
Yeah, thanks for bringing that up. Another question, Ken, how do you handle indels?
I don't like indels. So out of all of the candidates that we've validated, I would say, out of all of the candidates that we've looked at, probably about half of the ones that turned out to be sequencing artifacts were caused by an insertion deletion that we found when we came back and done the Sanger sequencing. Out of all of the de novo SNPs that we've found, which is probably somewhere on the order of 40 to 50, we've only found one de novo insertion deletion, and that was a 3 base pair insertion that we found in the child. So in our experience, the insertion deletions are not very common in de novo SNPs, and they do cause a lot of false positives.
Okay. Let's see. Another question. Let's see. Is the software applicable for larger case control studies? Yes. We can handle massive amount of data, and that also correlates well with another question about handling full genome sequence data. We can handle that too. Can you analyze paired normal tumor seq data for somatic mutations and apply similar filters? Ken, do you want to comment on that at all? I've got a little bit to say, but I'd like to hear what you think.
So I don't see any reason why you couldn't. I have not done that. One of the things that we have looked at are identical twins where one of them has a phenotype and one of them doesn't. Again, I think you just really have to define what the mechanism of the variant that you're looking for, and figure out what are the features of that particular type of polymorphism, and then design the filters for it. I don't, I've never done that, but I know with the homozygous or the monozygotic twins that we've looked at, it's not been an issue.
Excellent. Well, I think that's about all the questions we have for today. There's a couple other real specific ones that I'll send to you, Ken, and we can follow up directly. The slides are available for download, and this webcast has been recorded and will be posted on our website as well. Again, Ken, we can't thank you enough. We also thank Cincinnati Children's Hospital Medical Center for taking the time to put this together. And again, if you're interested in applying this kind of technology to your research, let us know. When you close out of the webcast, there's a little survey. Check the box that you want to be contacted, or be more direct, be bold. Give us a call or reach out to us on our website. So thank you, Ken, and thanks everyone for joining today.